Thank you very much for the invitation. And I would like to talk about a new product from ZEISS. So it's the Correlative Cryo Workflow. One second here. Something not working. Can I reduce the images from the participants? Do you know that? Ah, okay. So as I said, I would like to introduce a new product from ZEISS, the Correlative Cryo Workflow. And cryo-microscopy has taken center stage in recent years, particularly in the structural biology community. Since award of the Nobel Prize in Chemistry in 2017 for developing cryo-electron microscopy for the high-resolution structure determination of biomolecules in solution, so the resolution revolution, as it is commonly known, has driven incredibly progress in understanding of the structure of isolated proteins, protein complexes, and molecules that could not be studied in any other way. With this molecular structure information to hand, the next key question is how that structure impacts the function of the molecule of interest in the context of the rest of the cell. And this question can be explored using cryo-electron tomography, so cryo-ET. Cryo-ET is achieved by acquiring a series of transmission EM images from a very thin sample, TM lamellar, which can be reconstructed to generate 3D protein density maps with resolutions at the submolecular to atomic level. This acquisition approach generates 3D high-resolution data sets of the molecule of interest in the natural context of the cell in which it is located. This is shown here on the slide. I have also some technical problems, it seems. So to reach the point of having a TEM lamella that contains precisely the molecule or structure of interest for analysis using cryo-ET, the most common approach is to first localize the molecule of interest using fluorescence microscopy. This is shown on the left side. Once the structure of interest is localized, the sample is then transferred to a FIPSAM, where the fluorescence information is used to localize the structure of interest inside the EM, and the TEM lamella can then be created prior to acquisition of the cryo-ET data using the TEM. The key for success in this approach is the precise high-resolution targeting of the location for generation of the lamella. An incorrect decision or bad sample quality both mean wasted time at the TEM, which is extremely expensive and inconvenient. Generating cryo samples that are of the right thickness and quality and contain the molecule you are interested in imaging has historically been a challenge. And this is the main reason that until recently, this approach has been limited to a small number of researchers globally. Each step of the process presents its own set of considerations and challenges, and these are laid out here in these lists. The most important challenges amongst these are generation of a high quality vitrified sample in the first place, maintenance of the sample in its vitrified state, creating images with sufficient resolution and contrast that the molecule of interest can be visualized using both the fluorescence microscope and the cryo-FIPSAM, then moving the sample from one microscope to another without losing or damaging it, and the precise identification of the location for lamellar generation to include the molecule of interest. The combination of these challenges have historically limited the reach of this approach since significant expertise has been required in order to generate high quality data from each lamella. The correlative cryo workflow from ZEISS simplifies this method tremendously, as you will see on the next slides. So starting with the first step of this microscope workflow, with the cryo sample prepared, the first step is acquisition of a light microscope image of the sample. The light microscope image provides both an overview image of the sample and the location of your molecule of interest using fluorescence. ZEISS has worked closely together with Lincoln to create an integrated cryo stage that fits to the Axio imager. The cryo stage provides exceptional stability. The cooling time is less than 15 minutes, so switching between room temperature and cryo modes is very quick. And then once down to temperatures, the system is very stable. So acquisition of data can be done for hours with no risk of devitrification, which is essential. Once your sample is mounted into your cryo stage, you need to acquire both an overview image of the whole grid, which is required for subsequent alignments in the FIBSEM, and high-resolution fluorescent images from the regions that contain the fluorescently labeled molecule you're interested in. An overview image of the whole grid can be quickly acquired using the 5x objective in transmitted light. The single-shot 5x overview is an excellent and quick way of navigating around the sample. From the 5x overview, the objective can be changed into the 10x, which provides a higher resolution image of the sample. Using this objective, the sample can be imaged using fluorescence to find the molecule of interest, or using reflection mode to look at the eye's thickness and general sample quality. Finally, having located the regions containing your molecule of interest, you can switch to the 100x objective for a high resolution transmitted light or fluorescence image of your sample that will be the basis of the targeted preparation of the TEM lamellar. Having the flexibility to switch to different objective lenses makes sample navigation and image acquisition very quick and easy. For the fluorescence acquisition of vitrified samples, the approach needs to be as gentle as possible because high laser power can result in devitrification of the sample. The other requirement is high resolution, since this enables more precise targeting of the location for lamellar generation, a key process for the efficiency of this workflow. The most robust way of achieving optical sectioning is using a confocal microscope. Traditional confocal acquisitions are done using standard PMT detectors, which provide enhanced resolution in X, Y, and Z by removing out of focus light using a pinhole. In general, setting the pinhole to one area unit is accepted to provide the best balance of resolution and signal. The Eriscan detector, however, is the next level in terms of image resolution and signal. This is a smart implementation of a unique detector that contains 32 distinct elements, each capturing 0.2 area units of light and together delivering 1.25 area units of signal. Taken together, these things mean that this detector delivers both higher resolution and higher sensitivity than the traditional confocal detectors. For cryo samples, this is particularly important as it means high resolution imaging without the need for higher laser power that would otherwise risk devitrification of the sample and compromise sample quality. So the Eriscan detector provides gentle, fast, and easy access to super resolution information. And this high resolution information is vital in the next steps of the cryo workflow when it comes to precise localization of the molecule of interest when the sample is moved to the FIBSEM. So having acquired an overview image and high resolution fluorescence data at the light microscope, the next step is to transfer the sample to the electron microscope. This has previously been a very risky part of the workflow, since moving between sample holders and instruments under cryo conditions risks losing or damaging the sample. The ZEISS Cryo Accessory Kit avoids all these issues because the sample remains in the same sample carrier for imaging using the light microscope during the transfer to the electron microscope and during acquisition of the FIB-SEM data in the crossbeam. The clever design of the sample carrier minimizes any risk of loss or damage to the sample, which significantly improves the overall efficiency of the whole workflow. The vitrified sample can be mounted on different sample carriers, such as temp grids, autogrid sapphire disc, or planchets used for high pressure freezing, and the cryo accessory kit provides attachments for all of these common sample carriers to maximize the flexibility of the users and the kind of samples that can be used. So with the light microscope data to hand and your sample transferred to the FIB-SEM, the next step is relocalizing the region that contains your molecule of interest. The ZEISS Crossbeam runs the same ZenConnect software as the light microscope, and this means straightforward alignment of the datasets once the sample is transferred to the crossbeam. The video shows some of the flexibility of the alignment tools to ensure the crossbeam is directed to precisely the right location as shown in the fluorescence image here. All of the data is kept in context, so no matter how many overview images, fluorescence datasets from different regions, SEM or crossbeam datasets are acquired, they are all maintained within the database structure of ZenConnect. This ensures no data is lost. It also makes alignment very easy, so the focus of the user can be on the structure of interest and the resulting images rather than the alignment requirements. So ZEISS crossbeams are designed to give highest usability and best contrast images. Imaging cryo samples demands low acceleration voltages to avoid charging, and usually this reduces the contrast to see the ultrastructure of cells and tissues. Even in this unstained state and when captured using low acceleration voltages. This is shown in the image, which has cellular components are clearly distinguished from their surroundings, making it possible that from the iris scan is also shown in the overlay. So the crucial thing about targeting cryo-ET is generating the lamellar at precisely the right location in all dimensions. Using the fluorescence data as a map to reach the right region, the precise lamellar location can only be captured if you can see the structure in the image that is generated by the FIB-SEM. This video shows the process of targeted lamellar creation, and you can see the locations either side of the sample are milled using the FIB-SEM. By visualization, the structure of interest during this milling process, you can clearly define the lamella in the right location and ensure that it contains your structure of interest, which is essential. So without sufficient contrast in the image, it is impossible to see the structure of interest, and then the location of the lamella needs to be done with guesswork. And that is nothing we want, right? So the movie shows the power of our crossbeam here. During the milling process, high contrast images are acquired. Remember, the sample is not stained at all, and the ZEISS crossbeam has the best imaging capability here. Cells and subcellular compartments are clearly visible, and the TEM lamellar location can be precisely selected. So, and on the right side at the end, the final TEM lamellar with a thickness of 256 nanometer is shown. So throughout the workflow, there's a requirement to carefully consider how to manage all of the data Capturing overview images, fluorescence images, SEM images, and FIP images from different instruments can quickly lead to a complex data management situation. And to avoid any data sets getting lost, ZenConnect keeps all of the data together and, most importantly, keeps it in context. So there is never a concern about losing any data sets in the workflow. This provides a lot of peace of mind and prevents many hassles at a later date with data stored in different folders with no reference to each other. Once the data has been acquired, there's a need to optimize it. For cryo-FIP data, both low contrast and striping are known considerations, and the ZEN-EM processing toolbox contains processing functionality to address both of these essential requirements. Having these tools conveniently available in ZEN saves a lot of complexity. There are two tools, these are two tools amongst many from the EM processing toolbox. Image visualization on the right side is another key part of the workflow, and FIP data in particular can generate large data sets that some software packages struggle with. The ZEN3D software module has been optimized to manage huge data sets with ease, and as such, visualization of 3D cross-film data is very straightforward and convenient. And at the end of my talk, I would like to summarize what I said with this movie. And with this, I would like to thank you very much for giving me the opportunity to introduce our new product, and thank you very much to the audience for listening.